Yes, we are ready.
Hello, everyone. Welcome to today's Introduction to Computational Structural Biology workshop. This is our second day, and I hope you enjoyed our yesterday's sessions, and we hope you would enjoy today's sessions as well. And I would like to show you today's schedule before starting. After my talk, Dr. Demet Akten will present, will give a theoretical class about molecular dynamics principles. And after a short break, we will have a practical session by Burak Servili, and it will be about molecular dynamics simulation. And after the lunchtime, we will have another practical hands-on session. It will be given by Sevilay Güleşen, and it will be about molecular dynamics simulation analysis. And then we will have a virtual party or networking session. And without further ado, I would like to announce next presenter, Dr. Demet Akten from Kadir Has University. Dr. Demet Akten, stage is yours.
Thank you, Batuhan, for this very nice presentation. So I will start my talk immediately. I will share my screen now. So thank you for all coming, first of all. Just a minute. Okay. So let me make it full view. Is everything visible? Okay. So I can start. Thank you again for inviting me to this wonderful organization. And I would like to talk about giant workers today in cells. And these are proteins and their dynamic structures. By the way, I'm from Molecular Biology and Genetics Department at Kadir Has University. So I'm happy. Sorry about the date of the that I put there. So let me just get rid of this. Okay. For some reason the bar is there. I cannot get rid of. Oh, okay. So I have to make it invisible, but for some reason. Giant molecules. So these are proteins that are essential components in all living organisms, and they are responsible for a wide range of biological functions. You can make the analogy of heavy laborers. You can call them laborers in our body, in our cells, that do a variety of functions for the survival of the organism. And these functions are simply, can be categorized and into many different types of functions. And one of them is defense. For example, antibodies are proteins and they have a certain shape. And moreover, communication, in communication department, we have the insulin, which regulate the sugar in our blood. And also enzymes are also proteins. They catalyze a variety of specific reactions. And moreover, we have the storage proteins in which, we have this ferritin, which store iron from our food. And more importantly, transport. Calcium pumps, which are embedded in the cell membrane, allows certain ions to move from cells to inside to the outside or vice versa. And therefore we can keep the concentration of certain ions fixed, and which is crucial for the survival, for the cell survival. And lastly, the structure, which is the collagen. Collagen forms the support of both the inner organs, cells, and also the skin. As you all know, we are able to keep the structure of anything, which can be a small entity like cell or larger tissues, through these collagen, which are also proteins. They are simply triple helices. They are flexible, and the lack of it, of course, as you all know, is one of the results of aging. Okay. Now, if you look at these protein 3D structures that we saw in the previous slide, we can see they are all different. But when we focus on the polymeric chain, because these are polymers, these are composed of similar units, they are all composed of the same unit, which is amino acid. And if we focus on the amino acid, we can see that the amino acid itself is composed of the same units, which is carboxyl group, amino, alpha carbon, and the side chain, which is different in different amino acids. And that's what makes the difference between one type of amino acid from the other. So we have 20 different side chains, which make 20 different amino acids. And these types are simply encoded in our gene, in our DNA. And these 4 letters AGCT simply determines what would be the type and the order in our body. And finally, when it is synthesized from the ribosome, it is simply built as a shapeless, set of beads. Each bead corresponds, each color corresponds to one type of amino acid, and they come together to make certain rearrangements within itself to finally give the 3-dimensional shape of the protein. This is called folding, okay? This order of the amino acid is what determines what would be the final 3-dimensional structure. So when we look at the order, DNA just determines the type of amino acid, the order of the amino acid determines the 3-dimensional structure, and finally, the structure itself is determined simply what would be the function of the protein, okay? So this folding problem is very important, how a protein gets its 3-dimensional shape in the first place. And if you look at the thermodynamic of this process, we can see that there's an energy difference, energy variation. And when there's this unfolded state, simply the protein has a shapeless, polymeric chain, and they do not have any function. They cannot function in this shape. They have to get into their 3-dimensional structure to be able to function in the cell. And while it is simply following the pathway through the native state, we call it, it just passes through some folding intermediates. And it is not just like the final shape, but it is just in between. It starts making certain types of interactions, and these interactions simply drops the energy of the system. And finally, when it reaches the native state, this becomes one of the lowest energy states it can reach. But sometimes things can go wrong, and these partially folded states cannot be successful in reaching that native final state. Sometimes chaperones can help, but sometimes in the absence of chaperones, they can simply go to a very amorphous aggregate state in which they stick together, they don't have any shape, they simply cannot free themselves from that amorphous aggregates unless there are some special chaperones around. But finally, they simply lock themselves together to form these fibrils, we call, or aggregates. And this is the main reason behind Alzheimer or Parkinson, because they accumulate in the neural system and they block simply the interaction between neurons. So this is a simple, very simple, short simulation, which lasts in 6 microseconds only for Willen headpiece. which starts from an unfolded state and tries to fold and find the final three-dimensional native state. Okay, and this is a very important computational experiment which can be conducted to predict the three-dimensional structure without going into the details of experiments, real experiments. So this is something very small, of course. We all know that proteins are much larger than that, are much bigger than this small protein, but this is a start. When you apply the same physical principles to larger proteins, finally you will succeed. So let me go back to this Richard Feynman, what Richard Feynman told us, the famous physicist, in one of his lecture series. It said, all things are made of atoms, and everything that living things can do can be understood in terms of the jigglings and wigglings of atoms. In other words, he means that everything fluctuates, everything is in motion. If you go deeper and deeper to look inside an atom bonded to another, you will see that there's a vibration, vibrational dynamics. And if we understand this vibrational dynamics, we will understand finally the function and the living organism. So what are these structural fluctuations and what caused them really? Well, simply thermal motion. So this thermal energy which gives this kinetic energy to the atoms in the first place. Okay, so when there's a frozen individual, you won't see anything vibrating. So when there's a thermal energy, that's what gives the motion. We are talking about thousands of proteins in a very small, even if it's a small biological system, we still have lots of atoms, thousands of atoms, which cannot be managed by simple quantum mechanical methods. We have to rely or generate more simplified models that will allow us to calculate certain phenomena in a reasonable time frame. And these are molecular mechanics. And in molecular mechanics, the motion of the electron is ignored. The atom is simply considered as a point in space, and the energy is calculated as a function of these positions of these atoms. In other words, nuclear positions. And when we do this simplification, we do a tremendous amount of simplification. And now we are ready, we can be studying thousands of atoms in a reasonable time frame. This is the function that you see, charm 22 force field, which describes the energy, the potential energy of the system, which can be decomposed as bonded and non-bonded terms. And I will explain to you the bonded part now. Now, as you all know, each atom is treated as a point with a certain charge, and they are connected to each other by simple harmonic forces, and they oscillate around a certain equilibrium distance. Also, if you think about the bond angle, these angles are also oscillating around 3 atoms, and we describe them by a harmonic potential, as you can see at the lower left side. On the right side, you can see rotations, torsional motions about some permissible, rotationally permissible single bonds. You cannot, rotate double bonds or aromatic bonds, but single bonds can rotate around themselves. And this is described by a cosine potential. And this is what gives actually the freedom for protein motion. But moreover, we have non-bonded terms. In other words, atoms which are not connected but close in space, therefore they interact, they see their effect from each other. How? Well, while short-range interactions, such as on the left side, this described by Leonard-Jones potential, we call it van der Waals interactions. And on the right side, non-bonded term, we see the Coulomb potential, the well-known Coulomb potential, which describes the interaction between charged atoms, either positive or positive negative. Okay, so this is the full potential, but we see the rotation or the motion of the protein, like an elastic material, due to the torsional degrees of freedom. And this is very important because as it rotates around that, it creates a new conformation, which is not different from the previous one. Actually, it is different in terms of conformational state. It's in a different shape. However, there's no bond breakage. There's nothing that has changed. No atoms added or no atoms missing. Everything is the same. All the atoms are connected in a similar way, except that they are simply found in a different angle, okay, different twisted way, or you can consider it like that. So these are like rubber materials, exactly, elastic materials that can perform or that can display a variety of different motions, shapes. It has different shapes, but the way they move are also distinct, are also specific to the protein. Now, these larger amplitudes, we can see bond vibrations in a much smaller time scale, like 1 femtosecond, but we see these types of larger amplitude of motions in a much larger scales, like 1 nanosecond up to 1 second, okay? So the time range is quite different. So we have another important property here, which can be embedded between structure and function. Why? Because dynamics of a protein is determined by the shape of the protein. And finally, the dynamics itself will dictate what the protein, what type of function the protein will have. So everything is depending on each other, and there's a close relationship. So we have to understand the dynamics. One powerful tool that you will also see after my lecture is molecular dynamics simulation. It's a methodology that allows you to see the protein in motion. Why? Because it allows you to create multiple or successive conformations of the same protein, and they follow the physical, most fundamental physical law of Newton's equation of motion, second law. Force acting on an atom is equal to its mass times acceleration. And everything is actually derived from there. And all the equations of motions are decomposed and rewritten based on your potential function that we described by Charn 22. So F is also first derivative of the potential, which when we combine it becomes this partial differential equation. You have to solve it. How? You have to solve, you cannot solve it analytically, because this R, the position, is not only the position of one atom, but thousands of atoms. So there's no way you can, solve it analytically. The only solution is numerical. Finite difference method is one of them. You have to break down the integration into many small time steps, okay? And these time steps is around 1 femtosecond. It can be increased to 2 femtoseconds. In many common molecular dynamic simulations, we use 2 femtosecond. So this Verlet algorithm, this finite difference method we call Verlet algorithm, was developed back in 1967, but it's still widely used. It's still implemented in all MD simulations. It allows you to calculate the position and the velocity for the next step, for the next consecutive step. But here, the most important or most time-consuming part, when you set the position and when you define a velocity, initial velocity, you have to come to these equations. To be able to calculate them, you need to know the acceleration. And in order to know the acceleration, you need to make sure that you calculate the force, okay? Force acting on each atom will only give you the acceleration. And the force is simply the first derivative of the potential function. So you have to go back to the potential, derive it, and first derivative and calculate the force acting on each atom. Why is it time consuming? Because we are talking about thousands of atoms. So we have to make that calculation many times. And once we know the acceleration, we can calculate using these two equations, the position and the velocity at the next time step. And then we are in a loop now where we can calculate all consecutive, successive conformations, positions, and velocity. And every time we come back to the beginning of that loop, we have to make sure that we calculate the force again, because now atoms have changed their position. They interact now in a different way at different distances. Therefore, the force is always updated. And that's the most time-consuming step of all molecular dynamics. And that's why we cannot go up to nowadays 1 second, maybe in very fast computers, but we are around 1 millisecond. And this is the best scenario if you have a very powerful computer. In your regular laptops, maybe you can reach 1 nanosecond. For 1 microsecond, you have to wait longer, a few months maybe. But with a 28 CPU server, just like this one, and you can, if you're working on a system, of course, it will depend on your system size. If it's between 80,000 and 100,000 atoms for 1 microsecond, the time required is 3 months approximately. And yeah, it's not happening in an instant. So it depends on your computer, depends on your system, and many more factors. I will share with you one of my simulation results that I did years ago. It belongs to a GPCR. It's a membrane protein, and it has allowed me to go deeper into the details of preparing a membrane protein system, because it's quite different from a cytoplasmic protein, because you need to consider the lipid environment, etc. You have to embed your protein inside, and it requires some sophisticated tools. So I will briefly describe this. What's the function, first of all? It's a signaling protein. In other words, something attaches from outside cell, and it just simply changes its conformation, and this is reflected to the G protein, which is coupled with our system of interest. So that's why it's called GPCR, G protein coupled receptor, because it's coupling to another protein, which is G, and then this G protein goes and continues this signaling to other proteins. It's the beginning of everything. You can think of it. So this is how it looks like when you extract that 3D structure from Protein Data Bank. And it has many different entries. It's one of them, but it's a huge protein. If you think about it with this G protein, it's a huge protein. It belongs to the GPCR, this subfamily, one of the families. We call it Rhodopsin. But we can see this other members. They are very similar, structurally similar, but they function in different ways. What are these functions? Visual sense, tasting, smell, also hearing, all sorts of visual other senses. And behavioral and mood regulation is also another function, another type of function, which is performed by serotonin receptor. And the immune system, they have certain important roles in inflammation and immune system activity. Also some growth and metastasis of some tumors. They are involved in a certain way. And finally, the most important ones, which is under beta-adrenergic receptor, blood pressure regulation and muscle relaxation, also digestive processes. And the one underlined muscle relaxation is performed by beta-2-AR, and that was the protein which I was studying. And these are the results of beta-2-AR I will share with you. Even though the function is so different from each other, and they have important functions, each and every one of these family members have its own specific function. Despite that, they have a same type of mechanism for signal transduction. So they activate in a similar way. They pass that signal to other proteins in a similar way, as this illustration shows you. So the green one is the GPCR, is our protein. And when an agonist is bind from outside, it can be anything. It can be a light, it can be an other molecule, or it can be something like a hormone, like epinephrine, for example, adrenaline. And any small molecule can attach itself from outside. And when it does, a G protein nearby comes and attaches itself, because now our protein becomes active. In an active state, it can attach itself to G protein. When G protein attaches itself, certain conformational changes happening, and they simply dissociate into many different units. So it has this largest unit, alpha unit, and it separates itself, attaches to another effector protein, and the other beta gamma subunits, the smaller ones, attach themselves to a calcium channel, for example. And when they finish with their function, when they finish with coupling with other proteins, they come back and reunite to form the G protein again. And that G protein now is ready to be used for the activation or for the cycle of another signaling. Okay, so when this happening, all these proteins, different proteins attached, have a role of transmitting that signal. And in the end, you observe this smooth muscle relaxation, or you can observe other biological effects, responses like smelling or tasting, or for example, heartbeat suddenly starts to have that effect on your heart, on the contractility of the heart. Unfortunately, in asthma patients, this cycle doesn't work properly. In other words, when an agonist binds, simply it doesn't activate the receptor. The receptor simply does not respond in a normal way. When that happens, a drug molecule called Ventolin, which is widely used, attaches itself to this receptor, binds more effectively than an agonist would bind, and simply starts activating it in a normal way. And as long as you take this drug, your smooth muscle will relax in this normal way, and you will not have difficulty in breathing. Unfortunately, these patients have to be careful. Why? Because it has a side effect. The side effect is as follows. Same beta-2-AR also exists in our heart muscles. And when you activate it too much, that drug will also go to your heart tissues and will activate the beta-2-AR in those tissues. And this will increase in turn the heart rate, the contractility. And as long as you don't have any heart problem, this will not cause anything. However, in case you have problem, this will turn into a serious side effect, and you have to be really careful in adjusting the doses. So when we go back to the GPCR family, we said all structures are similar, three-dimensional structures. Yes, when we align them, when we just put them on top of each other, we can see that they consist of a very specific 7TM structural motif, alpha helices, 7 of them, and combined in a regular way, parallel to each other. However, when you look at the sequence similarity, you don't see much. And on the other hand, when you look at the mechanism of signal transduction, you see exactly the same way things happening, the process of transmission of the signal. So even though the conservation at the level of sequence is low, the structure and the mechanism of signal transduction are the two things that have been evolutionarily conserved. And these are the most important parts of this structure. This 7TM, as you can see, so you have a barrel-like structure in which there's a cavity formed on top and also at the bottom, at the intracellular side. So this is where the G protein binds itself. And this binding cavity holds the small molecule. So you can see here, a small molecule simply binds to this upper level, extracellular side. And this signal propagation or the changes in the helical conformation affects the lower or the intracellular binding cavity from which the G protein will attach. And the signal propagates just like this through the conformational variations. Okay, so the dynamic structure allows this signaling to reach its destination. Now, this is the one of my studies, simulation studies for this protein, in which we created two different models. In simulations, you can do anything. It's so not possible. It's impossible to do the same thing in experiments. But in simulations on your computer, you can create whatever you want. And this is what we did. We just took the one model of beta 2 AR, this we call it loop model, and we simply extracted or get rid of that loop structure, and we clip the two loose ends together to create a second alternative, which we called clip model. And we want to see here, we perform two different simulations, and we want to see how this removal of this intracellular loop will affect the final dynamics of the protein, how the protein will react to the absence of this loop region. Because we know we want to emphasize the importance of intracellular loop 3 on the overall dynamics and the function of the protein, which was not something that was discussed in recent years. So this is how it looks like during simulation, the loop model. And you can see when I go back to the beginning, you can see how this yellow highlighted loop region just wanders around a little freely. It has a higher mobility than the rest of the structure. And finally, it just locks itself to the bottom of the receptor and stays there and lose almost all its mobility. And it's as mobile as the rest of the protein now. But in the beginning, it wasn't like that. Suddenly, it locks itself. So there's a transition here to a very different conformational state that was not observed in any experimental structure. So we didn't see this in any X-ray or NMR. This is something we call it this closed state, a very inactive conformation of the protein. Why we call it inactive, I will come to that later. But before that, I want to also say through a couple of figures, just like that, monitoring the distances and everything, we saw an important characteristic of this protein, which was already known, allosteric coupling between the extracellular and intracellular parts of this receptor. So there's a connection in between the dynamics. When the loop region just simply closes itself under the receptor, the upper part just opened up, expanded, in which the binding cavity becomes much larger. So as a result of that. So everything is correlated, and we can see this through the graphs. And let's focus on this upper part. What's happening there? Well, at the initial state, you see the loose end and the loose region of this ICL3. And finally, getting into shape and closing, and also forming some more helical conformations. We call it loop because it doesn't have any certain helical or some other secondary structure states. It's just a loop. But as you can see, as it closes itself, it's also forming some local helical structures. And finally, at the end, we see a locked state, a very inactive state, for which there's no way we can observe a GPCR coupled to a G protein. So this large G protein has no way, has no entrance for being able to couple itself or bind itself to the cavity under the receptor. This is how it would activate. And now, in these circumstances, there's no way for activation when we simply obtain a very inactive, locked state. We can make this analogy as follows. We can model it as a pincer-like behavior. You can think of the receptor either as a pincer or a bouquet of roses. Why? Because when you squeeze the bottom, the upper part opens up. And when you do the opposite, when you squeeze the top, the bottom will open up. It's a pincer-like behavior, okay? Which was already known, but which was not observed in a simulation where the ICL3 was involved. And this is due to the motion of the ICL3, which just derives this type of allosteric coupling, okay? And our new inactive state was proposed as a new target site, because now we have a very different conformation of the binding site, which is much more extended than the previous conformations. And the expanded size allows to maybe screen different drug candidates, which would be much larger in size than previously, because it will fit into that into that binding cavity more easily. Okay, so it's always better or more practical to try different conformations when you perform a screening. Okay, when you try to screen and find a novel drug candidate, it is much more advisable to try alternative conformations for your protein. After that, we did a second work. We continued on this receptor allosteric coupling, and I was interested in allostery for many years now. And at that time, I found a different approach for monitoring this allosteric coupling in a much more reasonable or time frame. I did several experiments, not only one simulation, but 9 different simulations, in which I tried to put some bond restraints at certain sites of the receptor in the binding cavity, so that I can easily observe these ICL3 closures in a much faster way. So instead of waiting like 600 nanoseconds, which took me like 3 months, I can design experiments now in a very, like 100 nanoseconds, which completes within a few weeks. And I was observing the same phenomena, same ICL3 closure, which is part of the allosteric intrinsic dynamics of this protein. So ICL3 is always closing, but is also opening again, closing and opening. So there's a continuous process in here. However, one of the handicaps of MD simulation is that when you lock to a certain state, your structure is locked to a certain state, and when it binds strongly with hydrogen bonds, as highlighted here with dot lines. Sometimes the number of these hydrogen bonds is too high, becomes too high, as in our state, as in our case. And there is no way we can free that state and go back to the original open state. Why? Because you have to break too many bonds. And the system cannot find that enough energy, to break these bonds and to go to its original state. So one of the handicaps is that when you find yourself, when your protein finds itself in a very low energy state, it simply cannot escape. The only way to escape that is to apply external forces. And during MD simulation, you can interfere. You can simply implement certain forces acting on certain atoms that you decide, and then just simply pull the system to its original state. But however, it is not what what we desire. Of course, we would see this phenomena by itself, but unfortunately, MD simulation simply does not allow that. In real life, this protein go back and forth. This ICL3 opens and closes itself, and it can never be in a locked state just like this. But in simulations, this is something we observe quite often. And this is why, but we collaborated with also Boğaziçi University. Two students, my two MS students were there, part of the TÜBİTAK project. Nowadays, it's going to be finalized next year, beginning of next year, but it has been going on throughout pandemic. My project, which is about identifying the regions, specific species-specific regions in glycolytic enzymes. And this was one of the exciting studies I did recently. I want to share with you some of the results, not too much, because I know that maybe you will have questions later. I want to leave enough time for that as well. Now, glycolysis is an important biological phenomenon, pathway in our body that allows for the production of energy. So glucose is metabolized. In the end, more ATP molecules appear in our body. So throughout this process, we have 10 different reactions, 10 different chemical reactions catalyzed by 10 different enzymes. We call them glycolytic pathway, and the enzymes are glycolytic enzymes. And we choose 3 of them: phosphofructokinase, glyceraldehyde triphosphate dehydrogenase, and pyruvate kinase. Why? Because those were the 3 enzymes that demonstrated some allosteric property, allosteric features. We were interested more in designing allosteric drugs. And people nowadays in computer-aided drug discovery area started to, switch to design or to target allosteric sites instead of orthosteric sites, because the allosteric site allows you more variability. Orthosteric site is more evolutionarily conserved, but allosteric sites are less conserved, and from species to species, you can identify differences. And as a result of that, you can just design certain drugs that will bind to certain species, but not to other species. Okay. That was the goal of our project. And those are the 3 enzymes, large, very big enzymes composed of tetramers. Here we don't have a lipid environment. However, we have a very large molecule instead. And they are all homotetramers, same subunit. came together to form the final active state. I will share with you the phosphofructokinase, the top one. And this is my group members, some of my MS students and professors from Istanbul Technical University and Marmara University, and also Boğaziçi University. We all came together. So our first paper, our first results were published in this paper as a new computational approach we developed. First of all, I have to tell you that we didn't just simply write the code, but simply combined different approaches together to find a consensus between them. One was energy-based, solvent mapping. In solvent mapping, you just dock billions of molecules to identify hotspots. Okay, maybe you've heard. These are low energy states. In other words, if you bind something to that state, this will have the lowest energy minima with your when it associates with the ligand or with the drug molecule. Therefore, they are candidates for target sites. On the other hand, we have this ENM-based residue scanning, in which we identify sites or residues in which, upon adding extra nodes to this selected residue, because in this model, elastic network model, we have a very simplified actually representation of the residues. Each residue is represented by a single bead and connected by a harmonic spring. And we don't have any extra nodes, extra atoms to represent that. However, during residue scanning, and this is something we developed by simply adding extra nodes as if it belongs to side chain atoms to each and every residue one at a time, and monitor the change in the essential or the collective modes eigenvalue. And we simply take the difference before and after. Before changing it, how it was, the eigenvalue, and after changing it, we calculate again, we take the difference, and we simply obtain a percentage change in the eigenvalue. And we simply detect the sites which have the highest change, highest effect on the eigenvalue. When we combine hotspots, and when we combine ENM-based residue scanning, this is what we achieve in the end. These are the ones with the lowest energy and also the highest frequency shift. Okay, and for phosphofructokinase, for pyruvate kinase and the third enzyme, as highlighted, as circled in these pictures, we identified very important allosteric sites next to the known experimental binding sites. And we want to make this comparison. How our proposed site have this allosteric effect as opposed to the known experimental binding sites or allosteric sites. And there was a tool out there which we all know now, AlloSigma, maybe you've heard, a very powerful tool. First of all, let me tell you, to quantify the allosteric effect of a ligand binding, but not only ligand binding, but also mutation. When you mutate a residue, how this mutation will affect or will propagate throughout the receptor and will affect the dynamics of the other sides of the receptor. This is what allostery is all about. When you bind something at a certain site, how this propagates, how the effect it reaches to the distant regions in the protein. Okay, this is what the allostery is all about. And then when you find a very effective allosteric site, you have the control over the orthosteric site as well, and this is what we want to monitor. When a ligand binds there to the proposed site, we observed negative delta Gs. Delta G is an allosteric potential, which is written in terms of lowest frequency normal modes. And this decrease in the delta G simply indicates a reduced dynamics. And we saw these reduced dynamics in almost all substrate binding sites. In other words, orthosteric binding sites. And more importantly, or surprisingly, let's say, we did not see the same decrease, same delta G decrease when we bind to the experimentally non-allosteric site. And the non-allosteric site didn't display any allosteric effect according to these results, according to delta Gs. And this is something that we did not expect to see that from the non-allosteric site. We were, hoping to see this also from the non-allosteric side. However, we didn't, and that was a surprising finding for us. But we were very happy to see the reduced dynamics that was displayed by the proposed site, by experiment number 1. And this was something very promising for us, for all of us. And finally, we came together and proposed or established this screening. FDA molecules, world not FDA, and also some more molecules from our experimental group at Marmara. And we combined this 7,200 molecule and docked to each and every proposed and experimentally known allosteric sites for each and every enzyme. And finally, we merged them. And we obtained, as a result, a total of 55 molecules which bind to each and every site effectively to all enzymes. And this was important because these are very promising, the top candidates, which bind as effectively as any enzyme in this list and in any region in that enzyme. And this is the final list, and we are simply in the process of measuring the binding affinity on wet lab. The experiments are still going on, and we have few promising results. Hopefully, we will be publishing that as well. Now we are back to the molecular dynamics simulation because we want to explore more. We want to explore. We want to make sure that these proposed allosteric sites do have an effect on the dynamics of the protein, and these simulations is the only way we can figure that out. If you are not going to perform any experiment, this is the only way, and this is the most accurate way, let's say, most reliable way to obtain that. So what we did, as we did for GPCR, we implied our bond restraints to these proposed sites. We simply fixated them, fixated certain bond distances. These are imaginary bonds. When I say bond, don't think about it as a covalent bond. These are simply distances between selected pair of residues, which become fixated throughout the simulation. So you cannot simply move them around. You cannot just see the motion. Those residues will be just like a ligand bind. Imagine a ligand is bound there. When a ligand is bound, it makes certain interactions with its neighboring residues. And during that interaction, it just fixates those residues for a limited amount. It's the same thing for our bond restraint. Instead of ligand, we simply implemented those bond restraints to mimic a ligand binding. Why we didn't want to bind any ligand, you might ask? Because the parameterization is something which requires quantum mechanical calculations, and it may not be 100% accurate all the time, but bond restraint is a very practical approach. You simply define the residues and you start simulation without making any major changes to your setup. So one way of avoiding ligand parameterization is to go through bond restraints, in my opinion. Why? Because there is a difference between apo state, which there's no restraints, and the restraint runs, in which we have these bond distances. Before and after. Well, this plot is not an RMSF profile, but the change in RMSF. So there's a difference. We didn't plot the two RMSF together, but we simply took the difference. And the negative ones belongs to the proposed site, meaning after implementing those restraints at the proposed site, the RMSF value at the first part, half of the tetramer at least, which corresponds to one dimer. In one dimer, the mobility decreased. There's a reduced dynamics in one mobility in one dimer of our tetramer. On the other hand, when we put the same restraint, same type of restraints, at the experimentally non-allosteric site, we see these magenta, the peaks that are on the positive side, meaning the mobility increased. Just the opposite. So we expect with an allosteric inhibitor, if this acts as an inhibition, we need to observe a reduced dynamics. And this is what we observed for our proposed allosteric site. And we were happy with that. And we go one step ahead and calculated now the fluctuation, not of the atomic positions, but now the distance between each pair of atoms. This value is very important because it also gives you the commute time, and which also corresponds to the amount of signal transmission between residue pairs. Here, the important part of this signal transmission is to observe, to highlight the pair of residues that are distant from each other in the protein, but they communicate. So they have a low fluctuation, distance fluctuation, meaning they transmit signal to each other, but they are distant to each other. And that can be a very important sign for a certain type of allosteric communication or signaling network. Okay, so if you look at two figures, one on the left side belongs to the upper run and with no restraint. And we see that the transmission or the signaling is mostly limited to each chain, and there's no, chain to chain or dimer to dimer transmission. But on the right side, when you look at the restraint runs, you see these blocks, these green blocks, the intensity increasing, the amount of residue pairs having a certain amount of distance fluctuation, less than 0.5, increases. This indicates that within each dimer, there is an increase in the signal transmission, signal propagation. So this might this might be interpreted in terms of allosteric inhibition. So upon going from active to inactive state, so up or run represent an intermediate state. In other words, up or run can sample both active and inactive states. However, in restraint runs, you are expecting to see something closer to the inactive. And this inactive state in the restraint run will indicate more connectivity, higher connectivity, higher signaling propagation within each dimer. And this is something that is not mentioned in any paper, in any research paper related to that. So there are not many MD simulations for phosphofructokinase, and experimental findings cannot point that detail for you. And this is only through simulations that you can figure out these detailed differences, okay, between the two systems. And finally, from principal component analysis, when you decompose and you identify these lowest frequency modes and calculate this correlation map for positional fluctuations, of course, which will simply indicate the positional fluctuations, positively correlated and negatively correlated ones are separated, as you can see over here. In orange color, you see positive correlation. In magenta color, you see negative correlations. And I highlighted some of the important ones, which I saw some differences between APO and restraint runs. For example, the red region simply disappeared in restraint runs. And those were between two domains, A1 and D2, from different chains. And similarly, other changes in other blocks also happened. And this is very important in terms of bond restraints, in terms of the effect on the vibrational dynamics, and also the allosteric itself is described here as a change in vibrational dynamics, not necessarily a big conformational change. Not all allosteries should display a large conformational changes in the protein. It can affect it at the level of vibrational dynamics, which can be only observed through an MD simulation and after principal component analysis. And I think this is, I'm coming to the end. Hopefully, I will try to sum everything. So we will continue. Our work continues with pyruvate kinase, glyceraldehyde 3-phosphate dehydrogenase. We can conduct more experiments for them as well to identify these allosteric regions. Finally, my final remarks about molecular dynamics is that I find it very valuable as a powerful tool, and it will simply record the protein motion, just like a video camera. And there's no wet lab experimental setup which can show this much detail to you within a few femtoseconds, which is, by the way, 1/4 of a second. Okay, it's so slow. And I found it also a very valuable tool for drug design studies as well. And that was my final remarks. I want to thank you all again for listening and for this lovely invitation. I'm happy to be here.
Thank you very much for your very nice presentation. If you have any questions, you can type into the chat box. Actually, now we have two questions. First one is that you mentioned that the force is taken as the negative gradient of energy. If you remember correctly, it's true for conservative force field. So are we considering the atoms as charged neutral after we are neglect the motion of the electrons?
Yes. So we are neglecting the motion of the electron, exactly. We are considering it as a point in space, and this is the set of components, energy components we consider. So when we take the potential, first derivative of the potential function, we are here, somewhere here, yeah, negative. And the position of each atom is rewritten in terms of the position of the nucleus. There's only one single point that represents the whole atom. Actually, atoms are complex. However, in this molecular mechanics model, atoms are simple points in space. I hope this answer helps.
We have another question. Are there situations where we introduce perturbation terms in energy to account for different conditions that might affect the total energy of the molecule inside the biological cell?
So outside forces, you mean, can you read the question again? Outside forces can be implemented. Well, if necessary, these are called steered molecular dynamics. I think that's what you mean. And if we implement outside forces, we can force the system to, for example, fold into another different structure, or maybe to accelerate certain biological phenomena. But this will not be, may not be accurate for allowing us to observe the real phenomenon, because it is something forced coming out from outside. This is not what would normally take place in real life. However, for certain types of experiments, yes, we can accelerate certain phenomena and implement certain forces.
And another question is, are there any online server to conduct molecular dynamics simulation?
Online server, no, because it requires too many CPUs, and people, it would be a heavy load for a server outside to perform such experiments, because it doesn't take a few minutes or a few hours. It might take much longer than that. We are talking about days, months. So servers cannot handle it. But you can use universities' CPU clusters. For example, TÜBİTAK has a facility just like that. And you have to, but it's not a server, of course. It's not something using a web browser, something you can start your job. You have to prepare your system and combine the files, right? And archive it, compress it, and go to the server, connect, and start your calculations. And yeah.
And another question is, in allosteric discovery, do bond restraints show some kind of freeze effect and decrease dynamics some overall protein regions? Thanks for all the information.
Yeah, this is what we saw. Actually, bond restraints, it's not something that people tried that previously. I never see anything published that explains the allosteric effect or observe the allosteric effect via bond restraint. And that will be my first attempt, actually. And I'm now writing on the process of writing a paper. Hopefully, it will turn out good. And I'm expecting, I observed this change in the vibrational dynamics clearly, as I showed you in my last slides. And it is so obvious that there's a change in there. And it's the first time that I observed it in a molecular dynamics simulation. I did the same thing for GPCR, for beta 2 AR, in a different way. It was also, my aim there was to speed up the calculation, the speed up the dynamics. But here I wanted to observe, make sure that my proposed allosteric site really has as an allosteric effect, and really has an effect of reducing the dynamics. That I wanted to see, and hopefully I think I observed it, and hopefully will be a publication in a few months.
We have another question. It is what you have used for the identification of allosteric sites in glycolysis enzymes, or it was just a random docking that followed the MDS?
Yeah.
And yeah.
Sure. Let me ask you answer the first question. It wasn't so it was a new approach. You can just simply check out this paper, Identification of Alternative Allosteric Site, in which we talk about this new approach. We combined both energy-based and structural-based. Structural-based is normal mode analysis. We combine this together with the energy-based computational approach to identify hotspots, which are also the sites where you see the highest frequency shift. And this is something that is only explored and implemented in this work. And there's no other example which combine both energy-based and structural-based approach together. Okay, so that was, and moreover, we, in addition to the proposed site, we also combined another tool, another powerful tool to identify or to quantify the allosteric effect, which is called allosigma. Allosigma is developed by another group and at Singapore, and we are simply communicating with them as well. And also they also confirmed our proposed allosteric site with this low negative delta G values. And we are happy to see that.
And another question is it necessary to do a apoprotein dynamics for ligand protein dynamics?
In order to see the effect, yes. In order to investigate how the ligand binding would affect the dynamics, so this is what we want to see, we needed to perform the apo runs as well. We can only understand the effect only by looking at the difference between apo and ligand bound state.
Another question is, I want to know if a ligand is binding in a site which is somewhere very far from active site. How will I go for checking its allosteric sites? Some broad steps will be helpful.
So your allosteric site, in order to be talking about an allosteric site, of course, it has to be far away from the catalytic site. Okay, and how will I know that this is an allosteric site? That was the question, right? So there are different ways, different tools. One of them is allosigma. You can start by using that tool, but also the consensus tool is to identify hotspots if these hotspots coincide with the allosteric site that you have proposed. And moreover, you can perform an ENM-based residue scanning to the same region. If everything is satisfied, then you can talk about an allosteric site, yes. And in addition to that, there are many alternative tools out there which rely both on energy minimization and also network normal mode analysis. I highly recommend to use alternative options as well.
We have some other questions, but due to the time limitation, we have to end this session. Okay. So, Professor Doctor, thank you.
You are very free to write to my email address. Yeah, I can. I would like to respond via email.
Professor Doctor, thank you very, very much for your answers for the questions and your very nice presentation.
Thank you very much. Thank you for your lovely for this lovely invitation. I'm very happy to see this type of organizations coming from students. I highly encourage you to do more and more. Okay. Take care, everyone. I hope you enjoy the rest of the symposium. Thank you.
Thank you very much. And our next session will begin in 5 minutes.
Okay. Thank you. Bye.
Bye-bye. Hello, everyone. And now we will move to the hands-on session of today's workshop. Uh, hands-on session. This hands-on session will be given by Burak Servili, and it will be about MD simulations using NAMD. Before starting, I would like to thank Burak to prepare the video that I just showed you. It was prepared by Burak. It was very spectacular video, I think. Thank you very much for this very nice video. And now stage is yours.
Thank you, Batuhan. I'm happy for that. Hello, everyone. My name is Burak Servili, and I'm a PhD student at Kadir Has University. To talk about myself, I'm working on molecular dynamics simulations and drug design. And my thesis is about an ion receptor named NMDA. It's in nerve cells. This is a hot topic because if there is any structural problem with this ion receptor, it causes some neurological disease like Alzheimer's and Parkinson's disease. Well, that's me. So let's start. And I want to share my screen. Okay, first, we're gonna check an online tool. It's CharmGUI. And CharmGUI is actually the best web-based platform to build complex systems and prepare their input simulation protocols. This is the interface of CharmGUI, and you should log in to use this online tool. And you can see on the left side we have menu. And please, after the workshop, please check the subtitles, and you will see some tutorials, and you will see some tutorials video about the CharmGUI. But now we're gonna continue with input generator part and this one. Okay, now we have actually a lot of options for this part, but now, for example, you can use PDB reader part to read your PDB file, and ligand reader, ligand reader. But for today, we're going to continue with solution builder. For example, if you have any membrane protein, you can use membrane builder part, but this part will be more advanced for today. So we're going to continue with this one. And by the way, you can go CharmGUI with me, and you can follow my steps. Now, in this part, we can call any PDB ID from RCSP, or you can upload your PDB file to CharmGUI servers. And we're going to continue with these options. But if you want to, you can create empty waterbox with this option. But now we're going to call a PDB file from server. And for today, we're going to use 3LZV structure. This is will be our PDB ID. And when we check this structure, it's structure of nelfinavir resistance HIV1 protease, and it has a small molecule named darunavir. When we check this structure, it's homodimer actually. Now we can continue with this one. We're gonna check our structure is usually. Now we can see we have two chain, orange one and green one, and also this is darunavir. So we can check together what is darunavir. So I want to Google it. Maybe we can copy from there the name. Yeah, darunavir. So I want to continue with the direct bank link. So this one. So when we check what is darunavir, darunavir is the HIV protease inhibitor, and it's threatened of human HIV infection. So I guess this information is enough for our structure. Now I want to go back to the main part. Now we can go back. Perfect. Now I want to check this part. In our structure, we have a small molecule, darunavir, and also phosphatidine and acetate ion. Now we can call this PDB on charm GUI, and I want to copy this ID. Now we can paste it. Now on the right side, you can see we're gonna continue the next step. That's mean we will select our models or chains or another materials in our system. Now you can see the first one, our first chain, the orange one, and prop B, our second chain, the green one. When we check the hetero part, now you can see we have some ID on there, 017 and POH4, for example. But I want to compare this ID on PDB's website. Now again, we can check small molecules part, and you can see same ID. So 017, that's mean our small molecule. For today, we're gonna create two simulation, and first one will be APO, that's mean without ligand, and the second simulation will be halo, and we're gonna use, or we're gonna select ligand options. But now we will continue with these options. Now when I click the next step, we will manipulate our PDB file. Now we can check together. Now in this part we have a lot of options, but the first one is about start and end point about chain. And for prop A, first we can decide our start point and end point for the chains, and the N term will be this one, and C term it will be continue like that. By the way, we cannot disable this part because charm GUI or NAMD must know start and end point for the chain. And when we check the next option, preserve hydrogen coordinates, actually now we're gonna create a new simulation. So we will not enable this part. So we can continue with next one. And the mutation part. Now, in your structure, you can swap any amino acid to another one. Now we can check our structure sequence on PDB part and structure. Again, we want to go this part. Yeah. Now I hope you can see very well this part. Now you can see we have some sequence for our structure. For example, okay, I want to select this one, 22. Now you can see 22, it's alanine. So we can go back to charm GUI interface. Now I want to change, I want to select the 22. So you can see charm GUI automatically find it, and it's now alanine. Now time to swap this amino acid to another one. So you can select, for example, we can swap this alanine to methionine. And so if you want, you can add more mutation in your structure, and you can use this button, add mutation. So we can continue like that. And for example, 21, you can swap this into maybe Leu, we can select this one. But for this workshop, we will not use any mutation or structure. So I want to delete this part and we can disable this part. Okay, the next one, protonation state. Again, with charm GUI, you can apply any protonation to some amino acid on your structure. But how can I say? Today we're gonna use NAMD, or we're gonna learn charm GUI with basics. So this part will be more advanced. Now I want to disable this part, but you know, if you want, you can add more protonation state for some residues in this part, and every time you can select like this. Now this sulfate bond parts. If your structure has any disulfide bonds, actually charm GUI will find automatically and it will select, but sometimes it cannot find it, but you can select from there directly and it will be add the sulfide bonds to your structure. And next one, again, we will not use this one. Phosphorylation. So if you need any phosphorylation for your structure, again, you can select any residue on your chains like that, and you can add phosphorylation and more. Okay, now this is GPR anchor. So if you want, you can add any anchor in your complex or in your molecular system. What is GPR anchor? And I have an image for you. For example, this is our protein and it must be linked with membrane. So these options, you can add any GPR anchor for your chain, for your structure, and we can continue with options. But our protein, it's not need any anchor for this part. So again, we will not choose this one. Now, and the next part is about glycosylation. You can select this one. And when we check the other options, by the way, for today, we have a lot of features to use it, but we cannot check all of them. But if you need more information, you can use this question mark, and every time you can check this definition. But now I want to skip this part, but you know, if you want to add any lipid tail, you can use these options for peptide stapling. Every time you can enable this part, but I said I want to skip these options. Now, if you need any symmetry part, for example, you have one chain, but your main structure must be homotetramer, for example. So you can use symmetry options, and you will be create your symmetric molecular dynamic simulation with your structure. Now, on the right side, we can continue the next step, and we will generate our PDB file. Now we're gonna wait a few minutes, actually seconds. Okay, perfect. Now in this first part, we can check our structure, and I want to click this one. Oops, not this one. Yeah. This is our original PDB, and you know, first chain, second chain, and some ions, and this is Darnavir. But now, the next one, and you can see we have only 2 chains because we select only prep A and prep B. So we're gonna continue from there. Now, water box size. Now you can change, actually you can use specific water box size. For example, you can type here 50 for X, Y, and Z. But I want to continue with fit water box size to protein size because CharmGUI can find automatically the true area for your structure, and also it will be less atoms for our molecular dynamics simulation part. And also you can change water box type, but we can, I want to continue the rectangular part. And this is H distance for water box. You can change this, but 10 angstrom, it's enough. Now, time to add ions our system. So I want to use the Monte Carlo method to add ions our system, and you can check Monte Carlo method on YouTube. Actually, you will find very nice simulation animation about Monte Carlo. Now, in this part, you can add basic ion types like this one, phosphochloride and sodium chloride, or you can add more ion. Now we can check it together. For example, for alkali metals, you can use these options. And for example, in this part, you can use magnesium actually, and you have a lot of options to add any ions your system. As a last part, if you want, you can add anion type or polyatomic anions for your systems. But now I don't want to use this one, and in this part actually we have potassium chloride. Now I want to delete this one, and maybe we can continue with sodium chloride. Yeah, now you can change ions in your system like that. Now when we check this concentration part, this is actually millimolar. So our in systems 0.15 millimolar ions, it will be in our system. But first of all, we should calculate all ions for our system. Now we can see we have 42 sodium and 48 chlorine for our system, but actually these are these values can change our simulation. Now we should continue to next step, and next step will be solvate molecule. Yep, now we can continue it. Now we're gonna wait a few minutes. Now Charm is creating our water box, and it's adding ions for our structure, for our molecular dynamics simulation. I mean, now we are preparing our system for to run it. Yes, it will be less than 2 minutes. Okay, perfect. Now our system, it's look like ready, but again, I want to check the first part. And this is our structure. Now we can check the next step. I want to reopen. Maybe we can check the first, this one. Now Changui cannot show this. Yes, perfect. Now you can see this is our water box. Now I want to check the full system with this button. Now you can see we have water box and these are ions and this is our structure. And now our system, it's look like ready. Now these values are about system size. Now the next options about periodic boundary. In this part, you can change these values for X, Y, and Z. And maybe you can use 19 for all system size. But as a default, I want to continue with the automatically part, and Changui can find all periodic boundary conditions. But now I cannot show this part to you. We will check this part on VMD, and I will show you how we can check periodic boundary parts. But now I'm just keep. Now time to continue next step, and again we're gonna wait a few minutes. I guess it's done, but now I'm waiting for website. Yes, perfect. Now, actually, the best part is here. Now, for force field options, you can select any version of charm, but the 36M, the last one. So I want to continue the last version of charm, and we will use last force field. And we need these options. And the best part it's here. For example, if you are using NAMD, you can generate your system files when you click this one, or you can used this one for GRAMACS, Amber, Genesis, Desmond, LAMMPS. But for today, we're going to use NAMD. So we need NAMD outputs. Now, another options. By the way, I said we're going to create 2 simulation for today, but all simulation has 2 different simulation part. The first one is equilibration, and the next one actually is production part. And when we check this part, now we can see we have only one options, NVT assemble. That's mean our simulation, in simulation, we should stabilize temperature and pressure for every time for during simulation. So we cannot change this part for equilibrium, but when we check the production part, we have the second option. And when we compare between NPT and NVT, actually in this option in these 2 options, temperature and pressure will be stable during molecular dynamics simulation, but with NPT options, volume can change. Now, actually, I have a little GIF for you. Now you can see the first one and last one, volume can change every time. But when you select NVT assemble, your volumes will not change for your molecular dynamics simulation. So we should continue with NPT for this part. As the last one, you can change temperature in this part. Now I don't want to change this value. I want to continue with the default settings, but you can change this value for your organism. For example, you can change this value. Now, when I click the next step part, I'm going to create our NAMD files. And actually, this is so important. We need input files to run NAMD part. When you check our GitHub link, you're gonna see these files, and you can download from there. Also, when you prepare your system in CharmGUI, you can continue with the download TGS button. Now you can download your file from there. Now, actually, this is your job ID. Every time you can use this link, this ID to reach your files. And now I don't want to download this file, but you can download it there. Now we can create, or we can go back. And again, we can start with input generator, and not this one, solution builder. We're gonna continue solution builder part. And again, we're gonna type here 3LZV, next part. Now we're gonna prepare our hello simulation. Now we should select this part. And again, we will not select the other parts. And this is our PDB manipulation options. Now you can see we have another options in there because I select our small molecule. That means we need some parameters or topology file for our structure, for our small molecule. That means for this part, you can upload SDF or mol2 file for this structure, for this small ligand, but in Charm GUI or in Charm GUI libraries, this Darinavir, it's in the database, so we can use Charm General Force field to generate this part, and we can continue with these options. But when you create a new ligand, you should upload SDF or mol2 files to create topology and parameter files for your small ligands. Then we're gonna continue with same options, and I don't want to select the other features. Now again, we can check our structures, and this is original PDB. But when we check the next hello version, now we can see Darinavir is here. So we can continue from there. And again, we're gonna create water bugs, and we will add ions for our structure. And you can see these files for topology and parameter files for our ligand. Okay. This is our GitHub link. You can download from there our files. Where is Charm GUI? And again, you should change this part. And I want to delete potassium chloride, and we can add sodium chloride. And we should press the calculate button and next. Now in this, for this step, I don't want to continue because we have time limit for this workshop, but you can continue in background, and we will not change any options again. Maybe we can wait only for this step. I want to show our water box with chain A and chain B. Also, we will see our small molecule in our system. Okay, now we can check this structure. Now you can see we have water box and ions in there. Now when we zoom in structure part, and you can see chain A, chain B, and Darinavir, it's here. Now I guess we can continue with NAMD part. So to start our MD simulation, you should download NAMD software from this part. And again, you can go there. But for this part, we have two options. The first one, by the way, we're gonna use the Linux operating system for run our MD simulation. But now we have two options. The first one, it's about CPU. So we will use, or we will run our simulation on only CPUs. The next option, it's about graphic cards. In your system, you can add graphic cards to run your simulation, and NAMD can reach NVIDIA CUDA cores to run. By the way, graphic cards more powerful than CPUs. So we have these options for in this simulation, but you know, I don't know your hardwares for today, and we're gonna continue with the CPU part. But if you have an NVIDIA graphic card, you can download these options, and I will show you how we can add or how we can run on graphic cards in this simulation. Now you can download the first one, and again, this is our link. Now I want to open into, yes, perfect. Now my folder is mtd. Oops. Perfect. Now this is our MD file, so we should extract these files or uncompress. So we're gonna use tar command. Actually, I don't recommend these laptops for MD simulations. You should use desktop or workstation. But for small simulation, we can use laptops. But how can I say, the cooling system is so important, because when we run an MD simulation, our CPUs will work fully, and we cannot control the temperature for CPU. So I don't recommend the laptops or mobile device for MD simulation. But for today, if you want, you can use. Now we're gonna extract our files with tar command. Tar, the options X, we have, that's mean create please new file and extract all files. This is the tar options. Now MD7. Okay. Now we extract our files. Now we can check it. This is our MD folder. Can you see it now? Actually, you can find these comments on this part. Tar, it's here for uncompress. Here. Now, can you see my screen? Is it clear? Okay. Now we're gonna go to an empty folder, and we're gonna check this folder. Now you can see we have an MD application, it's here. Now we should run this NAMD, but first of all, I want to use /NAMD2. So you can see we have error. We can see error in this part. It's about fatal error. So because NAMD needs a config file to start our simulation. Now I'm going to go back. Change my screen like that. Okay. Now we can go our folder, and I want to start with the with ligand part and without ligand part. With ligand, okay, for now. And I changed the folder name. Actually, you can do that. And this is our charm GUI. And I said you can download these files from there. And for the with ligand and without ligand part, and you will see same folders, same files in there. Now, when you enter the charm GUI folder, you're gonna see a NAMD folder. So we should continue with this one, NAMD. So we can check all files in this part. Now we can see we have coordinate file, and again for coordinate file, this is PDB file, and this is PSF file for structure, protein structure file. And you can see we have two input file. That means these are our config files. Now we're gonna use, or we're gonna call these config files with NAMD software. But first of all, I want to check these config files together. Now we should use less command to check these files. Okay. The first line, it's about calling structure or coordinates files, and you can see step 3 input PCF, or I want to show these files. Oops. Step 2 input. Yeah. These two files. We're gonna call these two files to start our simulation. The next one, it's about temperature. Maybe you can remember this value on charm GUI. So you can change this value after charm GUI steps. Now, the next comment, it's about creating new files, and new files will name with step 4 equilibration. We can change this part, but I want to continue with the default settings. And we need parameters file, and we're going to use source command and name define this file in folder. And you know, I didn't write any comment because charm GUI create these comments or these files automatically. So you can use charm GUI to create your config files, and it will be easy to create these files. Now the temperature command, so you can call with this symbol with this two value. So our main temperature will be this one, and again we set it the output name as step 4 equilibration. So we're gonna call this name with this command. Now next command, it's about first time step. And you know, we're gonna start our MD simulation, so we didn't do anything. So our start point will be 0. Now, the next comment, restart frag, DCD frag, and XEST frag. This comment, it's about frequency. That means you can see your, or you can restart your steps every 1 PCS. Then this is 500 equal 1 PCS. And for example, for output energies, you can type any value, this part, and every 125 steps, you're gonna see energy values for your structure. And also for timing part. Now we should continue the next part. Now you can see we have force field parameters. Now for our system, the first parameters files or this topology files, actually it's enough. But charmingly add all parameters files in your folder. And again, we can check together. Now you can see we have topper folder and all parameter files in there. And you can call parameters file with this command and namely find these files automatically. And you know, I said I cannot explain everything in this part because we have a lot of options, but you can check all features or these comments on MD user guides, and it's here. And again, I'm sharing this link on chat box, and you can find all parameters in this part. For example, MD configuration parameters for input part. Now you can see for coordinates, structure, parameters, and you can find a lot of options for your MD simulation. But for today, we're gonna continue with basics. So I don't want to change default settings. And also you can read this information or description for these comments. But in this part, time step, this is so important. And now this value is 2. We will check this time step on the next last line. And you can see we have constant temperature part. So every time temperature will be stable, it will not change. And maybe you can remember from Charmgui, and this is periodic boundary conditions, and we select automatic options, so you can see every time NAMD find this value. And this part, it's about water box, but basically we will not change any options for this part. And we will check PMA part in VMD, as usually we will check it. And I said constant temperature. So, you know, our temperature and our temperature and pressure will be stable. So you can use these options for these steps. But again, I don't want to change these options. Please check this website to learn all features. Now, in this part, we have constraint part, and this is so important for our MD simulations. Now we can see we have reference file, and you can find this file in folder. When you check this file, you're gonna see B column, and you can change B column with between 0 and 1 values. For example, when you type the B column 0, that means don't apply any constraint. But when you change the B column, it will be constrained. It will be apply constraint on any atoms. Now, how we can use this constraint part? In NAMD, we should apply constraint for first step with minimization part, because we cannot release all atoms at the same time. We should apply, for example, first of all, we can select backbone of structure and we can apply constraint. So first of all, side chains will move, or we will see side chains dynamics. Then we should release backbone or the another parts for your structure. So when you release all items or all atoms in your molecular dynamics simulation, you will see some crash because CPU cannot calculate the first step. So constraint part is so important. But you know, we use charm GUI, and I said charm GUI automatically find constraints steps. So we will not change anything for this part. But for example, if you have a large or huge protein structure, you should find some method to start your MD simulations. Now you can use this part to apply constraint in your structure. As the last two steps, we have a minimization part, so we can change this value. This is so important to find minimum energy. And num steps, this value will be our endpoint for our MD simulation. And run, this is so important. Now you can see we have value for 125,000. When you multiply with run value with time step, so maybe I can type here on checkbox time step, multiply run. Now you're gonna see your time of MD simulation. That means now we can multiply these two values, time step and run. So calculator, we can use it. 100 multiply 2. So that means it's femtosecond. 250,000 femtosecond. So when we convert this femtosecond to picosecond or nanosecond, we can check this table. Now you can see this is femtosecond part. So you should delete last 3 0 for to find picoseconds, and for this for nanoseconds. So divided by 100 to 250, picosecond, and again divide. So we have 0, 1, 25 nanoseconds for this simulation. I hope this part is clear. Now, time to run NAMD. Now, first of all, we should go back and we call NAMD software. And I hope, no, it's not here. By the way, this options, that's mean go back folder. Now you can see our NAMD file, it's here. NAMD folder, M7, yes. Now when we check this folder, not this one, we're gonna use CD to enter this folder. Now you can see our NAMD is here. Now, I want to run this NAMD with dot and slash. Actually, it's depend of your project, but I guess it must be more than 100, and it's acceptable value, I guess. But I said it's about your structure, because if your structure has 1000 amino acid, for example, more than 1000 amino acid, so you should change your run values. Maybe you can go 1 microsecond. Okay, now we can start this NAMD application like that. And again, I want to press enter. And again, we have an error. Actually, we can do NAMD simulation on Windows, but now you can see I'm using Windows 10, but now you can use Ubuntu or another Linux operating system on Windows, but you should activate, where is the our GitHub part? Yes, it's here. No, MD, not this one, this one. Now you should active VSL2. So when you type Google VSL2, you can follow, you can find a lot of options how we can active VSL2. But I checked how we can use NAMD2 on Windows 10, but it's not working very well. So I installed the Ubuntu part, so we can use NAMD with this method on Windows 10. Now again, I want to run this NAMD, so you can see we need config file. So time to show this config file NAMD2, but for this part we have some options to run NAMD, and the first one will be P options. P options so important because we will type to here our core number or core value. So first of all, we learn how we can check how we can check hardware of your computer. Now I want to type here CPU ls CPU. Now this is my CPU, and it has 8 core, mainly 8 core. But when we check this part, threads part, it's 2. That's mean in my CPU, the 8 core, it's main core, but when you multiply these 2 values, that's mean in this GPU, it has 16 core, and half of these cores it's virtual. So that's mean we can use all cores for our MD simulation. Now I want to continue our comment, so we can call NAMD2 plus P. That's mean, for example, when I type here 16, it will be use all cores of CPU, but I don't want to use these options because we can see some crash during Zoom session. Now I want to type here 2, but that means it will be very long. Now our second options plus, I want to check my notes. Yes, ISO malloc. When you type this, when you type isomalloc sync, that means we will not see any message from NAMD2, so we can get better performance with this command. So the next command or next options, set CPU affinity. So we can increase affinity of CPU. That means all cores will work synchron to each other, and we can use these options. Now, time to show input file for NAMD. So I want to go back for first folder. Now I want to check my folders. Yes. Now this is our folder, 3LZV. I want to continue with the with ligand folder. Halo part. CharmGUI folder, you can download this file. And you can see we have a lot of files, but we should go NAMD folder, and it's here, NAMD. Now you can see we have step 4. Where is step 4? Okay, step 4 equilibration input file. So we should call this config file for NAMD2. Now I want to type here larger than symbol. That means we need log file for this process. And again, I want to go back or for the main folder of our simulation. NAMD. And again, we will check all comments together. Now I want to change this part as a out. If you want, you can type here out or log file. But now I want to type here out. Now again, we can check our script. Now we can start NAMD2 application with this command /NAMD2 P plus P. That means we're gonna use 2 core of CPU. Isomalloc sync That means we will not see any message from NAMD2. So it will we may get more power performance from CPU. Set CPU affinity. That means all cores will work together, and also we will increase our affinity of CPU. Now the next command I want to select this part. Time to show input file to NAMD NAMD2 software, and larger than symbol. That means create our output file to here, and with this name. As the last one, I want to add and symbol. That means our NAMD2 software will work on background. Now time to press enter. Now you can see this is ID for NAMD2. Now I want to type here top. And actually you can see NAMD2, it's here, it's working. And process ID, this one. Now I want to Control C, you can quit or you can exit this part. And also I want to show another software command. It's htop. It's alternative of top command, and you can use sudo get install htop, so you can install this application on your Ubuntu or Linux part. Now we can call htop, and actually it's look like better. Now you can see I have 16 core and only 1/3 core is running. For NAMD, it's here. Or if you want to, we can kill this process. And I want to select this one, for example. Now you can press F9 to kill this process, and time to send kill signal. Now you can see now core is not working, and we cannot see any NAMD process in this part. Now I want to go back. Maybe I can open a new Ubuntu file. Yes. Oops. Now I can use this terminal. Again, I want to check the same folder from there. Folder, and every time you can go on NAMD folder when you download your targeze file from CharmGUI. Every time you can find config file in this part. Now I want to check output file. Now you can see we can check this part. We have some information about in this simulation, and this is our parameters file. Go down. Now in this part, every time you can find energy values, temperature average, pressure, volume. So you should learn how we can read log file. And actually, after my section, you will check this part with Sevilai, and I hope you can check this information from log file. And you know, I killed this process, so it will stop there. Okay, now I said we can use our graphic cards to run NAMD part, but now I cannot use CUDA cores because my graphic cards it's not in my desktop. But I want to show you how we can use these options. Actually, it's so important to get or to make fast your MD simulation. And I said graphic cards so important and so fast. Then CPU. Now again, we will use these lines, and again we can call MD2, or we can run MD2 software like that. Now go back, and I want to add new comments for this part. Plus device. So when you type here devices, sorry, with S, you should type here graphic card ID, like 0, or for example, if you have graphic cards more than 1, you should use comma like that. But how we can learn our graphic cards ID? This part important. Now, when you go the terminal, and you should type here NVIDIA SMI, but you can see I cannot show you because I said it's my graphic cards now. It's not in my desktop, but I have image. When you type NVIDIA SMI in your terminal, you're gonna see this screen. Now we can check together GPU name and fan temperature. Now in this part, where's the shapes? Oh, not this one. Yeah, this one. GPU name. Maybe we can change. Oops, not. No field. Yes, GPU name. So this is GPU name. Tesla P, another part, but graphic cards ID, this one. So you can see 0. So we should type 0 for this part. Now we can go back. Now I want to type here 0. So that's mean NAMD2 will use our GPU. But by the way, I want to copy these lines. We cannot use multi-core folder. We should use CUDA version. It's important. So tar xvf NAMD2 CUDA. Yes, CUDA version. Now we can use only with CUDA version these options. Okay. So as the last one, I'm gonna add last options for this part, plus idle power. Idle. Okay. Cool. Now that's mean our NAMD application every time will send signal to GPU or our graphic cards, and the NAMD will keep the GPU awake. So this is this part important. When you type device 0, you should add these options. It's so important. Now, when I type when I press enter, it must be run, but I said my graphic card's not in my desktop. But if you have any NVIDIA graphic cards, you can use these options, CUDA options. This is important to obtain more power. And also, I want to share this link with you so you can find a lot of options or a lot of feature for NMD command. Now you can see, for example, we can check this figure, CPU power for nanosecond per day. But when we compare with Tesla P100 graphic cards, and you can see actually differences. So graphic cards so powerful to NAMD, and actually it must be NVIDIA card. And you can see another options. And for example, we can check this part together. Not this part. Yes, now you can see, you will see some options like set CPU affinity, device options, and you should know device ID, and you can use NVIDIA SMI for these options. And when you type NVIDIA SMI, you will see this screen, and you can find device ID from there. Yes. I guess that's it. I hope it's clear. But if you have any question, I'm here. And I will try to explain it. But you can use NAMD2 with these options. By the way, if you don't have any desktop or workstation, you can use TÜBİTAK. Demet just said like that. Also, you can use Google Cloud. Okay, this is the server. You can use this platform. Or Amazon AWS, another service to run your molecular dynamics simulation. But you know, it's not cheap actually. But if you want, you can try it. And I said it's not cheap. And thank you for today. Do you have any question? Okay, can you please tell how to build a system where the box size is increased on extracellular side? Okay, actually, I want to show my presentation file. I guess this one. Yes. And I said this part actually more advanced, but we can go charm GUI together. Now, I want to continue with membrane builder part, and we will choose layer builder, this one. Now you can see in this part, now charm GUI will use OPM database, and you know, Jamil said OPM servers. That means charm GUI will call membrane information from OPM servers. For example, we cannot use this PDB ID for this one, but I want to show you. Now I want to click the next step, but if you want, you can create on your membrane. But when you choose these options, actually it cannot find, I'm sorry, it's my mistake, membrane builder. Now we will use this server. Yes, now I want to continue directly. Now you can see this part, generate PDB and orient molecule. That means we will create our membrane. By the way, I will have a question. As you show, if we use proteinate state for inside amino acid in the protein, will the result be so different from previous use? Yes, it will be different. And you can see in this part, you can use orientation options to put your protein on any membrane. And next step, and we will calculate selection area, but I want to skip this part quickly. And I guess we have 7 minutes as a last. Now you can see this part. You can create your lipid membrane, or you can change water thickness, and also you will see a lot of options to create in a membrane, but we cannot check all options. But you can see again we have box type in there. We can use rectangular, so you can create your membrane water box with these options. And you can change box size with this value, and you can add membrane for your system. Okay, now I want to show the another part, because the membrane builder part is so advanced. Now I want to change it. I want to continue next step. Next. And next. We will not change anything. Now again, I will show you how we can change box size in solution builder part. Yes, now, and can you see these options when you click specific water box size? Now you can type size for your system. Now you can like that. Now you can do like that. Actually, it will be in Charm PDB step. Do you have another question? I hope this workshop will help. It will be helpful for you in the future. I hope you can use this part in your project, and you can find this video on YouTube, on our RSG YouTube channel. And thank you for today.
Burak, thank you very much for your nice hands-on session.
Thank you, Batuhan.
And now we are moving to the lunch break.
You're welcome.
Afterwards, we will have another hands-on session by Sevilla Güleşen. Is there any other questions? Let me check YouTube too. Seems like we don't have any other questions, so we can move to the lunch break. See you at 1 p.m. GMT+3.
Okay, see you.